Hi, class, and welcome to probably what will be the final recording of the semester. This will be our last lab, and we'll go through cell division and development, and we'll get through part 1 on mitosis. So this is lab 16. I'm going to go ahead and share with you at the end kind of what you'll be expected for your lab exam. But for right now, I'll kind of take you a little bit through the lab guide, and then we'll go over what you'll need to kind of complete for the end of the semester assignments. So this part 1 will just go through mitosis. You'll look at the terminology of mitosis. You'll identify the different stages of mitosis. And the lab guide will take you through kind of an activity that we would be doing if we were in person. So here's the terminology that you should know. You don't have to define them, but you should know all of these terms because you'll be using them to label different structures as shown below. So as a part of the mitosis simulation, all cells come from other cells. And in most cases, new cells will be identical to old cells. And this is because genetic information is replicated or copied and then passed on to new daughter cells. Cells produce new cells by dividing, and thus one gives rise to a new cell, each identical to a parent cell. So mitosis refers to the division of the material in the nucleus after the DNA has been replicated. And the result of mitosis is always two identical nuclei, each having the identical numbers and types of chromosomes. Nuclear division is followed by cytokinesis, which is cell division, resulting in identical daughter cells that will be completely identical to the parents. So this simulated cell cycle materials, the lab guide is going to take you through what this lab simulation would have looked like if we had done it in person. And it'll take you through the different steps of mitosis: interphase, prophase, metaphase, anaphase, telophase, and then cytokinesis. And you're basically going to have to draw what you see with the proper labelings to each section. Then you'll observe mitosis in an animal cell slide that we provide for you. Use the lab guide images to locate and illustrate one cell in interphase, and then one cell each in the 4 stages of mitosis. Keep the stages listed in order, so draw an interphase cell first, a prophase cell, a metaphase cell, anaphase, and telophase. I really hope you've been over this in lecture. If not, I'll take you through the lab guide now, but you're basically describing or labeling the stage, starting interphase, prophase, metaphase, anaphase, and then drawing what you see. And be sure that you're labeling each phase in each cell with these structures listed next. And you can also put in the proper zoom magnification for that stage. Then you'll answer several questions about the activity we just did. So let's take you through the lab guide, because this will take you through exactly what you need to know. So here is the mitosis simulation, and in the classroom, these are the materials that we would have. And the materials as a part of the mitosis simulation, the yarn strings themselves represent DNA in the form of chromatin. So it's just tightly wound up DNA. And then these pop beads in red and yellow represent condensed DNA and visible chromosomes. So here is what we have. You have an unreplicated DNA in chromatin form. Then we have replicated DNA, 2 molecules of DNA. When we put the 2 kind of pop beads together, we have 2 molecules of DNA forming a chromatid. And if we separate one out, we have a daughter chromosome. So the following images will just help you better understand the model for this activity. And then this instructor has taken you through kind of what happens in each phase. So this is G1 phase of the cell cycle, normal growth that the cell is going through. S phase is another normal phase of the cell cycle before we get into division. And then we'll go through the mitosis phases. So in prophase, the nuclear envelope will start to dissolve. So we have drawn on a desk in lab a cell itself with the DNA in the form of chromosomes within the nucleus, and the nuclear envelope is starting to dissolve in the prophase. In metaphase, we can see that the DNA is starting to line up on what we call the equatorial plane. The spindle fibers are attached to the DNA strands because eventually those DNA strands will get pulled apart in the anaphase stage. So again, we're going through the stages of mitosis, which involves taking one cell and dividing it into two cells. In the telophase stage, the DNA will kind of turn back into its less dense chromatin form. As a cleavage furrow occurs, we start to see the forming of two new cells. And then cytokinesis literally means kind of splitting of the two cells. And now we have two daughter cells that have been formed from one. So you'll use those previous slides to basically draw exactly what you see on your worksheet while labeling each slide and each phase appropriately. In part 2, you'll be observing mitosis in an animal cell, and we give you the slides for looking at a whitefish blastula. If we're looking at a mitosis in an animal cell, we give you different kind of slices, and then we'll zoom in on one of those slices. Each of the next several pages, it's important to note, shows numerous cells in various stages of mitosis. So you'll use the images to locate one cell in each stage and then draw it. Be sure to label the parts as indicated on page 4 of your worksheet, and you can also use your lecture slides or your textbook to help. So for example, here's a look at a whitefish blastula slide, and we see lots of different cells here, and you see cells in different parts of mitosis. So for example, where my cursor is, this is a cell in metaphase because you see the DNA nicely lined up along the equatorial plane. Here is a cell where my cursor is in anaphase because you see the DNA being split apart. This might be a cell in prophase because you don't see a clearly defined nucleus. The nuclear envelope is starting to develop. And as you go through, you can use any slide you want to kind of locate the different cells in the different parts of the stages of mitosis. This is a really nice one in telophase because you start to see the cleavage furrow of the cell dividing into two. And let's see if we can find kind of a cytokinesis. This is a good look at metaphase again, lining up. I don't really see a good look at cytokinesis. Cytokinesis, again, would show kind of two cells right next to each other right after division. But you should know, they should be in here. This will be telophase. You can see the clear cleavage furrow. And then whenever you see kind of two cells, this would probably be a good example of cytokinesis because you see the nice long cell membrane dividing the two. So that takes us through kind of this week's final lab, lab 16. And I'm going to go to my most recent announcement just to kind of show you our final weeks of lab. We just have this week, and then next week your final lab exam will open. So for this week, you need to complete the quiz and the lab 16 worksheet. And then your final lab exam will open on Monday, December 6th. You have all week to take it. It will be due on Friday, December 10th. It will be using Proctorio, but I'll be using a lot of quiz questions from previous quizzes to make up this final lab exam. So you can use your previous quizzes to study for that. I think that brings us through kind of the end of the semester. This will be the last recording that I make. So thanks for bearing with us on another online semester. You guys have done great, and take care, and we'll see you soon. Let me know if you ever need anything or you need any help. So take care, guys.